We're in the final technical talk stage, and I'm really, really delighted to welcome Greg here. I guess now for the third time in a row, possibly more, I'm not sure. Thank you so much for joining, and please take it away.
Hello, my name is Greg. I'm a relatively new faculty. I just started my lab about 3 years ago at UC Berkeley. And I want to start with this slide. How many of you have seen this movie? Oh, not this movie, it's a TV series, right? Netflix series, Altered Carbon. Yeah, great. All my futurists seen it. So for those of you who haven't seen it, they basically achieve immortality. They have this cortical stack that records all your neural activity. And if, let's say, you die, your body, what they call a sleeve, can go, but you can put that stack into a new body and, experience life again in the same sleeve, or maybe a new person, different gender, anything you want. So how far are we from this future? I think what we need to enable this future, so you don't have to die, you can live forever, is molecular machines. And of course, we know proteins, they've showed us amazing molecular workers and other machines. So many people who doubt molecular nanotechnology, you are all a proof that molecular nanotechnology exists. We are all the result of many of these molecular machines diligently working at the same time. And that enables me to speak to you. But can we adapt them to do something useful for us besides just, by physical tools, maybe record neural activity? And some people say that, we already can create new molecular machines. I would say that they're mostly thermodynamical minimum based trained on X-rays. We cannot still create activated states and dynamic machines. So that's why when I looked for a field to join, I got attracted to DNA nanotechnology because it's just a little bit more programmable and understandable for me as a chemist, even though I started as a biologist and then turned chemist and I'm in the electrical engineering department at UC Berkeley. So of course, all of you know that DNA is a stack of base pairs, and typically it serves as a genetic molecule. And our DNA comes from our parents and defines who we are and controls protein synthesis. But in 2006, Paul Rodemont from Caltech came up with this brilliant idea of DNA origami, where you can use DNA molecule as a programmable scaffold to self-assemble any arbitrary shapes, almost arbitrary shapes, using DNA origami. So in DNA origami, you can take a long scaffold, buy a bunch of short staples from a DNA synthesis company, and they send it to you in a couple of days. You can weave this orange scaffold into a rectangle. You can make a smiley face, and here's individual base pairs forming. Bring in two parts of the scaffold together. Like in Foresight Institute, you've probably seen this scheme many times. When I saw the smiley face, I decided I got to join this field. This is a single molecule, about 100 nanometers. And so what's revolutionary here, you encode programming language into molecules. Go Bears. And during my postdoc, I created nanoscale Mona Lisa, scaling up the size of this smiley face about 100 times. And of course, Stella earlier today in the morning showed that she can do it even bigger now at Harvard. And we can make programmable combinatorial arrays. We can even play tic-tac-toe with DNA. So we can control reconfiguration. But these are all kind of fun and games. Can we build something useful with this? And that's what my lab is doing. And the very first PhD student who joined my lab 3 years ago was interested in neurodegenerative diseases. And he was very interested in monitoring activity of all these neurons in our body. And typically right now, if you want to get single neuron firing resolution, you have to insert this 1024 channel count electrodes, drill a hole, insert an electrode. Sorry about the graphic images, but just to show you that this is not very scalable. We have 100 billion neurons in our brain. We can only record maybe 300 at the same time. Can we do better? With molecular machines, I think we can. So he designed in just 1 year this DNA origami piston that has a stator that incorporates into membrane using cholesterol molecules that are hydrophobic, and a moving part. And so on a stator, you have certain boring nanoparticles that are now a little bit more exciting because we can use them as a signature in MRI. And you're, of course, all familiar with FRET, where you have fluorophore quencher when they're close by. Fluorescence is quenched. You don't see fluorescence when they're far away. You see fluorescence. Turns out there is a magnetic version of this. And of course, we want to use some form of readout of a neuron activity that can go through the skull, so we don't have to drill a hole in the skull. And our skull is not very transparent, so we cannot use fluorescence. We can use magnetic field. It goes unattenuated through the body. So there is a iron oxide and manganese oxide particles. They have different relaxivity, and basically you can create pulse sequences in your MRI instrument to read out what state your piston is in. So Benjamin Carey in our lab designed this machine, and he didn't have any idea how to work with DNA origami. He was an undergrad in X at MIT in power electronics and MEMS, and he learned DNA origami in 1 year. And just to show you how easy it is to kind of learn this particular way to make molecular machines. And so he made this. This is some negatively stained TM images. So we had to add some contrast bars so we can see the orientation. He inserted them into giant unilamellar vesicles, which are kind of artificial cells, and control the movement of this piston. And so it's very important not to have much of activation energy in this movement for fast actuation, because neurons fire really fast. You don't want your machine to be too slow. And so we had an actuation and control session just before this. We want actuation happen as fast as possible. Neuron fires more than 1 time per second, so it's really, really fast. And so he showed that we can, as soon as we activate this artificial neuron, we haven't even studied with real living neurons yet. Our fluorophore and quencher move, and we can see the different signals. These are not even magnetic particles yet, but soon they will be. All right. And here I'm going to tell you about kind of grander vision in our lab. 1 minute? OK, great. So we want to convert arbitrary biological signals into electronic signals, and the other way around. Not just electronic, but also chemical, mechanical. This is a kind of scheme. And 2 postdocs from my lab, Yunjeon and Soyeon, are here on this workshop. You can talk to them. So they developed the general approaches to maybe achieve this utopian vision. Thank you.
Awesome. We're combining our nanotech and our neurotech track.
This is certainly very interesting. I was wondering about how you get the data out and how much data there is. If the goal being 100 billion neurons, and they fire pretty quickly. So what sort of data rates do you expect this technology will be able to get out?
So with MRI, you can see a lot of different voxels, and for each voxel, you can have a value of T2 star relaxivity. So you can read out billions of neurons firing if you can achieve strong enough signal and low enough time for each signal. Yeah, existing infrastructure with MRI instrument that is basically in every hospital would allow us to get a lot of data.
I wonder if you can compare, for your engineered, let's say for the fluorescence-based version of your voltage sensor, how does the performance of that sensor compare to genetically engineered sensors that people like Michael Lin have developed, that are based on natural voltage sensing domains and fluorescent proteins?
Yeah, that's a great question. Protein sensors, of course, have a lot of advantages, is that you don't need to deliver them, you can just express. And Michael Lind's, the recent paper, I mean, I cannot compete with that yet with DNA origami. But that's fluorescence sensor, right? If only we could find some kind of light that goes through the skull. So we haven't compared, to answer your question, the thermodynamics and the speed yet. We're still trying to find a good way to characterize it because there is. So we're now trying to put this into kind of patch clamp so we can do single cell studies. And hopefully after that I can give you a more satisfying answer how it compares to best protein sensors.